All right, everybody. Thank you so much for coming today. It is my great pleasure to introduce my friend and good colleague, Dior Kelley. Dr. Kelley is an assistant professor at Iowa State. I will give you a bit of background. Dior did her undergrad majoring in chemistry at UC Santa Cruz, then got a PhD at UC Davis, working in plant biology department with Chuck Gasser, working on patterning and polarity in ovules. Then sticking with California, did her first postdoc at the Salk Institute, an NIH postdoc fellowship with Jeff Long, working on microRNA function during embryogenesis. And then after a short period there, switched to UCSD, HHMI with Mark Estelle, and that's actually where I know Dior from. We overlapped there. We were benchmates. Had a lot of fun times there back in the day. I'm sure I annoyed the crap out of her with my constant tapping on the bench and all that stuff.
Tapping.
I can't sit still.
Lots of tubes of seeds.
It was a mess.
Yeah.
But it worked out in the end.
It was fun. Yeah, we had fun.
And so that's where our shared love for Auxin signaling comes from. She worked on Auxin proteomics, and has continued along the Auxin pathway now when she moved to Iowa State University in 2015 as an adjunct. And then since 2019, she's been a tenure track assistant professor there. Yeah, still working on Auxin and development. And yeah, with that, please take it away. Looking forward to it.
Thank you for the intro. Stand over here. So thanks for the invite. It's been fun to meet a bunch of new people today, and I'm looking forward to some more awesome meetings this afternoon. Of course, CBUS. We have known each other for a long time. It's been a while. So yeah, I have been working on Auxin also since I was an undergrad. And I think it's just one of these things that's hard to get away from once you kind of get into it. So I'm going to talk today a little bit about some work that we've been doing that we recently published in the group, actually just last fall from one grad student who finished. And then the rest of the talk is going to be mostly unpublished data. My group works in both Arabidopsis and maize, studying how Auxin mediates control of growth processes, mostly in the root, also a little bit in the shoot. But I'm going to talk solely about our maize work today, because I feel like the crowd here is definitely a lot more kind of, crop kind of interested. All right, so I'm going to start by giving credit to the people that have done the work. A lot of our projects are collaborative in nature, and a lot of students have really contributed to all the data that I'm going to talk about today. So Lincoln, he's a genetics and genomics grad student with me, actually my first student, and he's going to be doing his thesis defense in like 2 months. So I'm very excited. Max is a grad student who just finished last fall. I co-advised him with my husband and longtime collaborator, Justin Wally. And then I'm also going to talk about work from Melissa Draves. She was a former undergrad in the group. She's now a grad student at Mizzou. And then of course, many collaborators. So Justin Wally, my husband and longtime collaborator, he can't say no when I want to do proteomics and other things. Brian Dilks, who's at Purdue for his quantitative genetics expertise. Alicia Schrader, I'll talk about some Auxin transport work today that we're doing with her, and Katie DeHesh, and then also Nick Lauder, who is gone now, and Miriam Lopez. So I also have a very talented field manager who is very helpful. We need to do lots of pollinations in the summer. And I want to say thanks to funding. Of course, like the research couldn't really happen without that. So most of this comes from my startup as well as USDA NIFA. Okay, so for those of you that are familiar with Auxin or try to forget about Auxin, it is a key hormone that regulates many aspects of plant growth and development. It also integrates with environmental signals for like disease and drought and other things. Naturally occurring auxins like indole 3-acetic acid, we tend to focus on in terms of how they can elicit phenotypic responses in plants, and also how auxin might be transported and metabolized, and how those things contribute to growth. Synthetic auxins are also kind of interest, mostly in the back of my brain, not in terms of like a reality. Like 2,4-D and dicamba, because they're used widely in agriculture and we still don't understand their modes of action. But we do know that they can elicit very similar changes in gene, transcriptional responses, differences in proteomic remodeling, things like that. In maize, we actually know not quite as much in terms of the genetic components of auxin signaling, auxin transport, auxin metabolism compared to other species like Arabidopsis, Marcantia, and Fiscometrium. But there are a few classical maize mutants, which have been described in the literature, that link auxin transport and signaling to growth and development. So here I'm showing these barren inflorescence mutants, the BIF1 and BIF4 mutants, which are really important for shoot architecture. You can see that they impact yield, tassel branching, things like that. And this mutant on the right is called RUM1, rootless undetectable meristem 1. This is an OXIA protein, so a signaling component for auxin. And you can see that in the absence of this transcriptional regulator, the roots are essentially devoid of all of those secondary branches, which are really important for like water and nutrient capture. So I kind of started working on auxin because when I moved to Iowa State, like they put something in the water. So you work on corn or soy or pigs. It's sort of inevitable, but it actually turns out that like, yeah, I don't know, being a longtime Arabidopsis lab rat, I do enjoy being out in the field, and genetics with maize is really fantastic. So it's a good system. So when we first started, we wanted to look at what sort of just phenotypic responses we could rapidly characterize in maize to use for both forward genetic screens, reverse genetic screens, and then also for doing profiling for looking at gene expression changes in maize. So if we take maize seedlings, we germinate them in a growth chamber, grow them for a couple days. We can either not treat them with auxin or we can treat them with auxin. And we get these very classical sorts of root responses that we're used to for Arabidopsis, like inhibition of the primary root. It also can inhibit seminal roots and do a bunch of other things. So if we do long studies where we treat like every 2 days for many days, you can see that there is a really remarkable change in root system architecture. So here we get that same inhibition of the primary root. In fact, the tip is actually somewhat agravitropic. We also see an increase in lateral root density. The root hairs are also increasing. That's kind of that fuzzy quality here on that picture. And then the seminal roots, which are also embryonic born roots, are inhibited as well by auxin. So we can do these synthetic treatments and see a lot of phenotypic changes. My group is really interested in figuring out what the molecular players are that kind of elicit these phenotypic changes and what the range of responses are in maize to auxin and some of the genetic drivers of these different processes. So mostly focused on the root, which is what entirely I'm going to talk about today. You can see notably, it doesn't do much to the shoot. So for those Arabidopsis aficionados, that Arabidopsis typically has a really strong elongation phenotype. You'll see that that's not the case here for corn. Okay, so we know a lot about auxin molecular nuclear signaling pathways. I'm not going to get into whatever the extranuclear, non-canonical, whatever stuff today. I'm not going to talk about that. But we know a lot of this from biochemical studies, genetic studies, and Arabidopsis, Marcantia, and Fiscometrium. So at the core of this pathway within the nucleus, we have perception of auxin, shown here is this little kind of yellow tooth, by co-receptor complexes, which are genetically encoded. There are many of these co-receptors. The tier AFB family of E3 ubiquitin ligases. There are 6 of these in Arabidopsis. These OXIA co-repressor transcription proteins. There's around 30 of these in Arabidopsis. So this perception event within the nucleus results in polyubiquitination and degradation of OXIAs, which is pretty quick. Most have a half-life of around 10 minutes. So when auxin comes into the cell, rapidly there's a change in proteome remodeling because in part of this protein degradation pathway. Now there's also another thing that's happening, and this is related to transcriptional changes, which are also very rapid. So we have these auxin response factors here, ARFs, 3 different flavors, A, B, C. There's been different nomenclature over the years. This is sort of the current nomenclature that are mostly physically interacting with some of these OXIAs and then either repressing or activating gene expression. So in response to auxin, we see these large changes both transcriptionally and post-transcriptionally in response to the hormone. And some of these things on the model are still to be sort of like worked out and are maybe a little confusing, and some ARFs don't really fall into this canonical pathway. But for the things I'm going to talk about today, they really fit into this sort of canonical class A ARF sort of pathway. So some of the questions that we're working on in the group are trying to really fill these knowledge gaps that we have in Maize. So this is a phylogeny from Andrea Gallavati's group of all of the ARFs, these auxin response factors in Maize and in Arabidopsis. So they're highly conserved. There's about equal numbers. ARFs have been studied by loss of function genetic analyses in Arabidopsis for a long time. So for many of them, we can link them to developmental processes. In Maize, we actually have no loss of function ARFs published at all to date. We are hoping to change that soon. But so a big question is, what are these different ARFs doing in Maize? They are highly conserved. Do they have overlap in their function? Do they have unique functions? Things like that. The other big question that we're working on trying to address is just kind of increase in our number of genetic regulators for root system architecture. So here is like a smattering of genes that are important for controlling the primary root, lateral roots, root hairs. We don't have a lot of genetic drivers that have been well described in Maize, and yet we know that roots are a really important aspect of plant growth and physiology, as well as things that impact like yield. So we want to just have more genes that we know that contribute to these different patterning processes in the root. And then the other outstanding question that we're working on is the role of auxin transport within the Maize root system. So there are many different types of proteins that can move auxin across membranes, pins, the ABCBs, laxes, auxes, and then these pills, these pin-like proteins that mostly reside in the ER. So today I'm going to talk about a couple reverse genetic and quantitative genetic screens that are sort of addressing all three of these questions together. If hopefully I can kind of get through everything. And I just wanted to mention kind of one other motivating factor is that, we still are lacking a lot of genetic understanding for how these different aspects of the crown root system, as well as fine scale patterning for things like root hairs, are really driven in Maize. And yet these are things where we can really make big impacts for things like water use efficiency, nutrient acquisition, things like that. And so many of these genes are actually from rice, and we know very few from Maize. And so some of these are related to auxin, like here we see some IAAs and yuccas for biosynthesis, but I think we still have like a lot more to get through here. Okay, so the first study I'm going to talk about was published actually just last fall. And this is a really straightforward experiment that we did to essentially catalog and characterize auxin responsive gene expression in the Maize root. So we did this in both like a temporal and a spatial sort of context. And so this was work from Max and Lincoln, and then two former undergrads in the group, Melissa and Michelle. So this is our overall approach. We took primary Maize roots that were 5 days old, which is shown here in this little cartoon. We hand dissected them into 4 different regions. We chopped off the meristems. We took the elongation zone. Within the mature zone of the root, we're able to peel away the steel from the cortical parenchyma and the epidermis. So we essentially got like 2 mature tissue types. And then these roots had been treated with either a solvent control or 10 micromolar IAA, endogenous form of auxin, for 30 minutes and 2 hours. So we picked those time points because they had been well characterized in Arabidopsis. So we thought we could do some comparisons there. And also we knew from marker gene expression that we were eliciting auxin responses in those time points. And then we took those different regions of the root, extracted RNA and did sequencing, and then looked for genes that were differentially regulated either across zones or across time points. And from this, we wanted to make these unsupervised gene regulatory networks, which I'll show you in a little bit. So looking at these regions of the root, you can see a couple patterns emerging right away. So within the meristem, we do not see a huge response to auxin. So very few genes, at least at this 30-minute and 2-hour time point, are auxin responsive in the meristem. This could be something that's due to just the chromatin state of these stem cells. Perhaps they're not very accessible to transcription factors, or it could be because maybe those cells are used to really high levels of auxin anyways. And so adding additional exogenous auxin doesn't really elicit a huge response. Now, if we look at some of these more differentiated regions of the root within the elongation zone, for instance, we can see that hundreds of genes are changing in response to auxin. And we can also see that there's a temporal context. So for instance, in the elongation zone, many more genes are auxin responsive at 2 hours compared to like the cortex and the stele, where we see a larger auxin response at 30 minutes at that shorter time point. If we take these genes and then we cluster them within each tissue type, the meristematic zone, elongation, cortex, and stele, and then we just order them based on auxin response, we can see that many of these genes are uniquely auxin responsive within a tissue or at a certain time point. So that's why we say that we're observing both temporal and spatial context for auxin responsive gene expression. Another way to look at this is by these upset plots, and most people find them a little upsetting, to be honest, but it's like a high-level Venn diagram. So if we take all of these different genes that are upregulated or downregulated within a tissue or across different time points and start to look for overlaps, we see a couple of unique patterns. So this is the 30-minute set of data, and this is the 2-hour set of data. Anything that is not shared across each of these rows is unique. So that means there are 346 genes that are unique to the stele that are auxin upregulated, as an example. Anything that has dots, which are connected between 2 of those rows, are shared genes. So those are shared transcripts. And we've color-coded them to indicate sort of the pattern of how those responsive genes are acting. So if you look at this group of 22 genes right here, 22 genes at T30 in the cortex, they're upregulated, and in the stele, they're actually repressed. So these are neighboring tissues, and that means that even auxin responsive gene expression across neighboring cells can be remarkably very different, right? In the complete opposite pattern. Anything that's green is in the same direction. So this now gives us a set of marker genes to look for auxin responses within a tissue type or within certain time points. And it also allows us to do some predictive network generation as well, which is what I'm going to get into next. Okay, so we did GO-term enrichment to see what sorts of things might be unique across tissues or across time points. Of course, we see auxin activated signaling as one of the enriched terms, so that's just sort of like a sanity check. But we also see that many genes related to GA metabolism are uniquely regulated in response to auxin, increased actually. And then we see genes that are required for cell cycle, cell division, and chromatin silencing are actually repressed in response to auxin in a tissue-specific manner. Okay, so now that we have all of these auxin responsive genes, we want to try to predict transcription factors that might be regulating them. And so we're doing this in silico using an unsupervised approach. This is based on Gene3, which is an algorithm for gene regulatory network reconstruction. So we take all of these differentially expressed gene products. So they could have been differentially expressed across tissues or across time points in response to auxin. And we have a big matrix there. And then we look in that data for all of the annotated transcription factors from Maize. So when we take those two things together and we put them into this tool, it's called Scion, which was recently published, then we can basically use that to predict these gene regulatory networks. So I'm not going to get into the clustering. There's lots of different ways to do clustering. I'm just going to kind of blow through this as well because it's been published. But we're trying to create these large networks, which we can then go back and functionally validate. So this is what our current gene regulatory network, which we visualized in Cytoscape, looks like for the auxin responsive genes in the root. So here what you're looking at is actually a really good representation of the transcription factors in Maize. So we have around 1,300 in the data set that we detected. Many of those are the Maize ARFs, so 27 out of 33. Which is pretty good. So that allows us to now start to make some connections between those ARFs that I talked about at the beginning and what their potential targets might be and how they might link auxin signaling together. So it's a very large network. Each of these circles also gives you a little bit of more information about targets that are shared across particular clusters or tissue types. So it turns out here, this biggest circle represents auxin responsive genes in the cortex and the stele, which are shared. And then there might be particular transcription factors within those that are regulating genes. Max also color coded this to add another layer of complexity for genes that are auxin responsive at either 30 minutes or 2 hours, or actually independent of time. All right, so we are most interested in connecting some of those ARFs back to function. And so this is sort of one of these ways to get at it. We go back and we look at that data, and something funky did happen to this slide with some of the color coding. So if we take all of the ARFs that we detected in our data, these are Maize ARFs, and we just look at their expression across these different regions of the root in response to auxin, we can see that many of them have very unique patterns. Some are auxin responsive, some are not, some are expressed in many regions of the root, some are expressed in only one region of the root. So we want to figure out what their targets are. So we go back to that big network and we pulled out all of the ARFs and made a bunch of subnetworks. And that's what you should be able to see here, but you can't. So each ARF is a pink node in the center. So that's the transcription factor. And then all of its predicted targets from our Scion network are these little dots, which appear to not be connected because the little gray lines are not showing up. On top of that, we have ordered these based on these class 8 ARFs. So those are annotated based on their structures and domains, the clade B and then C, and then these Etten-like ones, which are sort of weird because they lack a particular region of an ARF, which is normally known to associate with like oxi-A proteins. Okay, so the take-home message here, even though it's not entirely visible, is that most of these ARFs have each of their oh, I don't know what's going on now with that. I'll just let it go. Okay, so most have their own targets. They're not shared. Some ARFs across clades do have shared targets. So this would sort of like buck this conventional wisdom that these different sorts of ARFs have very unique targets. So for instance, here, like this ARF8, 25, ARF23, a lot of these targets are predicted to be shared between these ARFs. So I'm going to leave this story here for now. We just published this. We have a couple of these ARFs that we're doing reverse genetics on, but we really need to, tag a lot of these and be able to do some more in vivo assays to actually figure out of these predicted targets, how many of them are actually targets in vivo. We do have DNA affinity purification sequencing data for most of these ARFs from Andrea Gallavati. But that shows on the level of like thousands and thousands of targets. So we've got kind of two extremes here. We have a predicted network with very few targets, and then we have these really large scale genomic data. So we kind of need to start to stitch that up together. Okay, so ARF27 is a TF that we've been working on for a while, again, based on reverse genetics. And I'm going to tell you a little bit more about what we think its targets are and what it's doing in the maize root. So when I first started working on maize, I went back to this root atlas data that was published actually by Justin a few years ago when he was a postdoc with Steve Briggs, where they took these same regions of the root and did integrated transcriptome, proteome, and phosphoproteome profiling. They actually did this across 30 different regions of developing maize plant. And I looked for anything that was annotated in an auxin pathway in those different regions of the root. And then I pulled out what are called like eigengenes because they had done some WGCNA analysis on that. So anything that had a peak expression, you can't see this very well here, but these are root tissues. So anything that was an auxin pathway sort of gene, like an ARF or a putative transporter that had peak eigen expression in these root zones, I got insertion lines from the transposon collection and started doing reverse genetics with these. And that's how we came to find that ARF27 is important for root growth and development. This is just from the uniform U collection of transposon alleles. Okay, I'm going to skip that because I just explained it. So ARF27, can we move that little black box, or is it? I didn't see it on there. Okay, I'll just keep going. So we have two transposon alleles for ARF27, one of these auxin response factors. And both alleles have a really striking phenotype, thank you, with respect to root hair formation in the elongation zone. So you can see that the root hairs are very sparse and they're very, very short. Now, if we treat these seedlings with Auxin, we also noticed another remarkable phenotype where normally within the meristematic zone, of course, these are stem cells, they're not differentiating into root hairs. But if we treat with Auxin, these mutant alleles ectopically form root hairs in the meristematic zone. ARF27 is the ortholog of ARF7 and 19 in Arabidopsis, and Gloria Madej's group has also been looking at Auxin responsive phenotypes in those mutants, and also sees this kind of ectopic root hair formation that can be titrated actually with increasing levels of Auxin. So we think this TF is really important for root hair growth. Maybe also initiation, because it does seem somewhat sparse. Root hair formation in maize is not via the same sort of canonical pathway as it is in Arabidopsis. And so there's a lot more plasticity. Many more epidermal cells can just become root hairs. So this also tells us that, you know, there might be some other factors involved too for just whether or not a cell decides to become a root hair or stay as a non-root hair. These mutants have other defects related to root growth and development. So here at the top is a graph of primary root length. You can see that both alleles have a short root. And if we treat them with Auxin, normally wild type is greatly inhibited in response to Auxin, but the percent inhibition that we see in both of these mutant alleles is diminished. So this implies that they're somewhat Auxin insensitive, which would make sense for an ARF that is no longer present, which is an activator ARF required for proper Auxin signaling kind of output for transcription. One of the alleles, the stronger of the two alleles, this -2 allele, also has a decrease in lateral root density. And when we treat with Auxin, normally that will increase lateral root density, mostly because it shortens the primary root. But we don't see any sort of Auxin responsive defects there with respect to lateral root formation. So ARF27 seems to be required for root hair formation and also for growth of the primary root. We don't know if that growth defect in the root is because of a reduced number of cells or if it's because of expansion or both. I get that question all the time and I still can't answer it, to be honest. Okay. We have looked at Auxin responsive gene expression in the ARF27 mutant. So we've done this using both transcriptomics and proteomics. We see that many genes that should normally be Auxin responsive, if you just take like this cluster at the top here, are really not Auxin responsive in the mutant anymore. We also see that there are a lot of interesting Go terms that imply crosstalk between hormone pathways, especially with respect to cytokinin in GA, and then there should also be oxylipins in JA as well. So we think that dysregulation of Auxin responsive gene expression by this ARF also is impacting other hormones. We haven't done any other hormone response assays on these mutants yet, but we're working on trying to look at some of those things in more detail. Okay, so ARF27 is a transcription factor. Most transcription factors are very well behaved in that they're in the nucleus, but some transcription factors can also be in the cytoplasm and either shuttle between the nucleus or be regulated in different ways. So we wanted to figure out what the direct targets were of ARF27. So we tagged it with a GS yellow fluorescent protein and expressed it in protoplasts. So we could do like chromatin IP assays and do some PCR, validate some of those targets. But what we started seeing initially was that this transcription factor was not in the nucleus. In fact, it was very much cytoplasmic. So this was not going to be super useful for us to do CHIP. And then Max started treating these protoplasts with Auxin. So he did a 10 micromolar 1 IAA treatment for an hour, and we started to see some nuclear accumulation in 3 hours, and it was very much more so in the nucleus. As a comparison, this is another transcription factor, MYC2, which is highly conserved, and you can see that it is solidly in the nucleus. So we actually have stable transgenic lines now for ARF27. It takes a while to do this in maize. So I think we're going to revisit these experiments, but we still probably will also do some imaging on those roots and make sure across these time points that the TF is actually in the nucleus for when we're going to go capture to do the chip to validate some of those targets. We might even do a couple time points. I'm not quite sure yet, but this behavior again is also very, in line with what Lucia Strader's group has seen for ARF7 and 19, those two paralogs of ARF27. So again, this implies that some of these behaviors of ARFs, even across species, are probably even for subcellular localization conserved. Okay, our current gene regulatory network from ARF27 from that transcriptomic data that I showed you just a minute ago that we put through Scion, that same network, has around 600 targets. So we're getting a little bit more than we got from our other network. We still have less than what we have for our DAP-seq data of ARF27, which probably has like 5,000 regions of the genome that are bound by this TF. But there's a couple that are really, really notable. And so RTCS1 is a well-characterized kind of like classical maize mutant, which has very strong impacts on root system architecture, is predicted to be a direct target of ARF27. We'd like to validate that. And then also we see RTH3 and RTH6, which are these root hairless genes. Also classical mutants are predicted to be targets here. So our next set of work on ARF27 is really going to be validating some of these targets and linking that disexpression or misexpression maybe of the targets with the phenotypes, especially for like the root hairs, but then also seeing what's going on with maybe some of these other hormone pathways that we think might be impacted by ARF27. So jasmonic acid, gibberellin, and cytokinin. And Max just graduated, so he's at Corteva now. Okay, so I'm going to switch gears and move from transcription mediated by auxin to how auxin might be transported within the root and talk about these new putative transporters, PILS2, PILS6. It's now currently annotated as a PIN on the recent genome release for maize. It's always like flipping back and forth on genome annotation, but they are closely related. I'll show you in a phylogeny. And so this is work from Craig, a grad student in my group. He's a plant biology grad student. Ariel, she was a grad student with Lucia Strader. She's now doing, I think, patent law work, and then 2 undergrads, Melissa and Michelle. You'll see them a lot because they're rock stars. Okay, so we know that auxin accumulates asymmetrically in maize roots. So we've taken that same approach of primary root, dissecting it into the different regions, and then measuring indole-3 acetic acid levels, actually in collaboration with Katie DeHesh by LCMS. So you can see that we have the largest accumulation of auxin within steel. The meristem also has a lot of auxin, and then the elongation zone and cortex have relatively lower amounts of auxin. So there could be different ways to set up these asymmetrical patterns. It could be biosynthesis, if there's only, generation of auxin from certain tissues by like the yucca pathway, or it could be through transport, or it could be through both of those things. So I'll talk about transport today, not working on biosynthesis or metabolism. So these pin-like family of transporters, the PILs, are known to reside within the ER, and they're thought to actually impact the pool of available auxin for signaling within the nucleus through moving auxin across this membrane. The closely related pins, there are some pins that are also in the ER, and then there's other pins that are actually at the plasma membrane. So even within a cell, there can be transport of auxin. So here's a phylogeny of the annotated PILS proteins from Arabidopsis. They have the AT designation, and then also the corresponding maize paralogs. So within this clade down here, this is the PILS2-6 clade. We have 1 ortholog in maize and 1 ortholog in Arabidopsis. These 2 PILs were actually candidates from the reverse genetic screen because they turn out to be eigengenes within some of the root tissues. So we had been working on these through this reverse genetic screen, is how we kind of came to focus on these 2 rather than specifically only caring about PILS2 and 6 per se. Okay, so these are enriched in root tissues. Again, we have multiple transposon insertion alleles for both of these. In fact, we have another allele that's not indicated on the line here for 6. So these have short roots. Black is wild type, W22. These are the 2 different alleles without auxin. With auxin, and the PIL6 roots are also short as well. We have not made the 2-6 double yet. Hopefully this summer we will have that out in the field. If we grow them a little bit longer, so those were juvenile traits, we can also see that they impact crown roots. So you can see here for this pill 6-1, they have a very, very remarkably different crown root structure compared to W22. So they're actually somewhat pleiotropic. They're short, they're a little bit chlorotic. So I think that they probably are expressed in many different parts of the plant. And overall, they reduce the stature, they reduce the size. So this was actually a little bit surprising to us because the pills 6 and 2 mutants in Arabidopsis, which have been published by Jürgen Feinman, are actually big. So it's like the opposite phenotype of what we were expecting. We also see some defects related to lateral root initiation. So here, Craig, these look like little nudibranchs, but they're actually maize roots that are stained. He did Flugen staining on these, so you can see these little purple pinky dots are where lateral roots are initiating and then outgrowing. And so he did staining on these 6-1 alleles. He also did 6-3, which is in a different genetic background. And you can see that there's a shift in the number of lateral roots that are forming. So they have the capacity to form them, but it's definitely at a diminished capacity compared to wild type. We've also looked at proteins and transcripts. This is super pixelated, which are misexpressed in the mutant. So here you're looking at proteomics data. We've also done phosphoproteomics on the mutant. So we have all of these different proteins, which are essentially dysregulated now in the mutant. And Craig has taken that data to try to figure out what proteins might be co-expressed with pill 6. Perhaps at the ER or just within the cell or within a tissue. So some of these modules here, so this is from a weighted gene correlation network analysis that he did with that proteomics data, like this blue module. This should actually be like more like a teal module, but it's like a lime green. Anyways, pill 6 is within these, and there's some other interesting proteins there that we're working on trying to figure out whether or not they're directly regulating maybe the activity of pills or modifying it, or actually being important for other aspects of hormone signaling. Okay, it's also ER localized, just like its Arabidopsis counterpart. So this is an ER marker here, this HDL marker, and you can see nice overlap between both of these, and this is in Nicotiana benthamiana cells. We do have stable lines now for pill 6 and maize, but I don't have any images of those yet. Okay, so then we also wanted to know whether or not loss of these genes impacts auxin accumulation in maize roots, with the hypothesis that they're transporters or facilitators of auxin movement. So Ariel did these assays where she took tritiated IAA, corn seedlings of different genotypes, wild type, pill 2 and pill 6 mutants, and then she did that same dissection, which I've mentioned before, into these different regions and measured levels of auxin, radio labeled auxin, within each region of the root. And so what we have seen, and she's done these assays actually a few times now with other alleles as well, is that there are defects in transport in the meristematic zone as well as in the elongation zone. So for pill 6, it seems to impact transport within those 2 regions of the root. For pills 2, it seems to be really specific within the elongation zone. So we see increased levels of auxin, which would be consistent with that efflux that would be required to get it out. It's basically being trapped within those regions of the root in this assay, because these carriers aren't there to move it out. Ariel has also expressed both pills 2 and pill 6 in yeast to see if it can transport across another heterologous system on a membrane. And so again, this is an incubation assay, and we see that expression of at least pills 2 here, I'm not showing the pill 6 data, can impact transport of IAA across the membrane. So we do think that these are bona fide transporters at this point. And this is our current kind of synthesis of both the ARF27 and pills 2-6 story that I just mentioned to you with our working models. So we know that ARF27 is an auxin responsive transcript itself, and it is required for root hair formation, and it's also required for root elongation. It could also be required for cell division because the roots are shorter, but this is our working model in terms of like what genes we think might actually mediate that. We don't know, but we have some targets to test. And then we also think that pills 2 and pill 6 are important for setting up these asymmetric gradients of auxin within the root, namely within the elongation zone to the meristematic zone. We have not visualized auxin you know, in a sort of in vivo way with like a sensor or something like that in the mutants. That takes a little bit more time, but we do have the D2 Venus sensor, we have the DR5 sensor, so those are being introgressed into these mutant backgrounds, and I think we should be able to do that really nicely, like on the light sheet. Confocal on maize roots is kind of a pain in the ass because they're really big. But that will be coming down the pipeline. So we would predict that there might be some tissue specificity there for how these things are modulated. Okay, so in the last little 10 minutes, I'm going to talk about a totally different approach that has kind of taken us to some of these same genes, but has also really opened up some other avenues. So this is a quantitative genetics approach to look at, you know, can we identify key genetic drivers of auxin-mediated phenotypes in maize roots? And maize has amazing genetic resources, including this Wisconsin diversity panel, which has around, I think around 900 lines in the total collection. We're working with a subset of 627. So these represent a lot of phenotypic as well as genotypic diversity, and they've been very well characterized. So we have lots of SNP data. They've been run for many other GWAS studies for things like drought. And so we bulked up these a couple years ago, right before the derecho hit in Iowa. And then we have been screening through them for different phenotypes. So I'm going to talk about the GWAS for Auxin. So this was work done by Becca and Melissa. They were amazing undergrads. They did this during the pandemic, sort of their pandemic project. So we had 627 genotypes. They grew them out. So for each genotype, we had 10 kernels that were planted for 2 different treatments. And we allowed seedlings to germinate. We removed anything that had not germinated so that everything was very synchronous because we were going to be scoring for root traits. At 3 days after germination, which was like scored, we started treating with Auxin. We did 10 micromolar IAA, that same concentration that we had already worked with before that we knew elicited a response. And then at 7 days, we imaged all the seedlings. And then we harvested the root tissue. So all of the root tissue went to the -80 for each genotype in order for us to do expression analysis. We've actually done transcriptomes now for 200 of these. We haven't gotten to the proteome yet. It's going to take a while. Then from those images, Becca and Melissa, in collaboration with Raj, who's a postdoc with Brian Dilks, did a genome-wide association study. So I will get into that now. We basically plotted all of the traits that were hand annotated in image J by Becca, Melissa, and a couple other undergrads. So here we're just looking at the root data. We also looked at shoot data. So this is the distribution of all of these different genotypes, 627 genotypes without Auxin treatment. The data is actually very well normalized. It looks really great. Now we treat with Auxin. Again, we get this canonical inhibition of the root across many genotypes. So that's working well. This is sort of centered here around B73, which is our reference genome. And then we also calculated the ratio of IAA to MOC. So again, we usually see an inhibition around 75%. And so this gives us immediately a baseline, but then we can see that there's a number of genotypes which are responding in very different ways. And we can also see that there is a huge range, obviously, in root length in general in this panel, which is not too surprising, but it's great because we have a lot of phenotypic diversity. Okay, so for GWAS, we followed kind of a standard pipeline. We started with around 20 million SNPs, which were from publicly available data already on this Wisconsin diversity panel. Any SNP that had an association with either of those 3 traits, so root length without Auxin, root length with Auxin, or the ratio of Auxin to non-treated within the root. So we did this for 3 different traits. Anything that had a SNP association at a p-value of 10 to the -4 or lower, we kept. So we go from a lot of SNPs to still a lot of SNPs because it's GWAS. And then we would define a locus in this method. And so there are different people that will do this different ways, but this is how we did it. So within a consecutive set of single nucleotide polymorphisms across a 250 kb window, we would say that is a locus that the SNP is residing in. And then we would take whatever SNP had the lowest p-value within that window and keep that one. So this is also one of the problems with GWAS is that our resolution is not great for like particular gene associations, especially because Maize is really big, but these are the windows that work well, given the size and annotation of the Maize genome. Then we would tag each of these SNPs to the closest annotated gene model. So this would be where the actual SNP was, but then we also take genes that are annotated downstream or upstream of that same position. So any of those are essentially candidate genes within that window. So from that, we got many positive hits. I think we have around 60 some odd candidate gene hits from this particular GWAS for these 3 traits. So each trait is color coded here. Root length with Auxin is yellow, mock treated is purple, and then the ratio is that kind of orangey color. So some of these I've highlighted because they are genes that we think probably are important for driving auxin mediated growth. So ARF27, that favorite TF that I already told you about, that's very important for root hairs as well as primary root length. ARF16, which is another class 8 ARF. ARF1, and then also scarecrow. And this is milkweed pod 1. This is a Kannadi transcription factor, which is also really important for patterning within lateral organ formation, not necessarily roots, but usually TFs are strong drivers of phenotypic responses. So these are just some of the genes that we're interested in, but we do have a lot of genetic drivers now that we could potentially either go back and try to validate with reverse genetics or see if we actually have available mutants from collections. So that is sort of what we did already. So each of these genotypes here, it's just listed K with a number. These are stocks of classical Maize mutants that we had a GWAS hit for, and we already had available seed, had not been screened for root phenotypes. So we grew them out. We treated them with Auxin, same 10 micromolar, or without Auxin. And we can see that a couple of these, the ones that are marked with very low p-values, do have an impact on primary root length, suggesting that these are in fact genetic drivers of root traits. And then we have a couple, like especially this mutant here, K18, which has a very altered Auxin response. So these are things that we're kind of starting to move forward. We're also going back and looking at some of our like candidate genes in terms of, are there pathways in there like ARF27, other genes that we know to be important as well for Auxin that we can test phenotypes for. Okay, so another way that we're kind of like trying to deal with this data on an unbiased way and not just, you know, picking genes that we want to look at is thinking about it in terms of, all right, a genotype context has a particular root system. How does that root system respond to Auxin, and or how does it behave in the field? So here I'm going back to that data showing the ratio of Auxin treated to non-Auxin treated. Normally we see inhibition of root growth. We have a bunch of outliers. These genotypes here are Auxin insensitive. So those are cool genotypes. And then we have these genotypes here, which are Auxin hypersensitive. So we took all those, we actually grew them in the field last summer. We let them go to V6, dug up all the crown roots, imaged them, and now we're working on extracting traits from those and trying to correlate and see if there's any patterns between the root systems that are Auxin hypersensitive versus insensitive compared to our reference genome. So I'm just going to show you a couple pictures of these from the field. I actually have a bioinformatics and computational biology undergrad who's doing his independent study doing image analysis on these. So I think it's going to take us the rest of the semester, but it's going to be worthwhile. So here are two of these genotypes that I just had marked for altered Auxin response. So B73, this is our reference genotype. And you can see that both of these have very, very different root system architectures compared to B73. So what we can go do now after we do these image analysis sorts of experiments on our crown roots is go back and look at like marker gene expression in these different genotypes. Are there things that we know are normally Auxin responsive, which are dysregulated in these mutants based on our transcriptomic data? Or are there other markers that are abnormally expressed? And these sorts of different root systems are also really important for assaying, ability to deal with drought, to capture different nutrients, especially nutrients that are at the surface of the soil versus like further down, phosphate versus like nitrogen. So there's a lot of things that I think we'll keep doing both in the field and then kind of getting back to genetically test these again to see where we can kind of push that a little bit further. All right, and so I would be happy to take questions. Thanks for your attention. I won't re-summarize these other things, but I'll just keep kind of this pathway up here and some of these other things in case you want to go back to them for questions. Thank you.
Questions?
Yes.
Nice talk. So you talk about the VTLSS, it's in the cytoplasm in the beginning, and then you have IAA, then goes to the nucleus.
For ARF27.
Okay.
Yeah, yeah.
So how that work,
I do not know. And I also don't know if it really is the same pool that's moving from the cytoplasm into the nucleus, or if there's active translation, and that is actually somehow now in response to this Auxin cue immediately going into the nucleus. What is in the cytoplasm is apparently, based on the Arabidopsis work of 7 and 19, part of these phase condensates. And so it's really not clear mechanistically, is it some sort of like modification on the ARF27 protein, like a phosphorylation or something else, which is mediating these condensates? Is it just these kind of like intrinsically disordered regions of the protein that are making it? So that's not clear. Lucia Strader's group is working on that actively with Arabidopsis. So we're not even still sure if it's the same pool moving or what. We don't know, to be honest.
Have you tried to express E. coli and see if it binds?
We haven't done any heterologous expression for 27.
If it's in a condensed.
If it's in a condensate or not.
Then it's probably it's hard to purify.
Yeah. Lucia's group has been working on like essentially sucking out with a needle the ARF7 and 19 condensates from Arabidopsis, and has been trying to do mass spectrometry actually with my husband Justin, and then doing some other kind of like biophysical experiments to try to figure out what else is in those condensates. So
Sounds like very difficult.
yeah. Yeah. Other questions? I don't know if there's anything in the chat.
I haven't seen any in the
Okay.
chat yet, but I can come up with a question.
No, Eric's got a question.
Oh, Eric, please.
Okay, is there a role for topics for the president?
Yes, it's called Ramosa Enhancer. So it's called REL2. And yes, it appears to be the exact same sort of association with oxiase and being a very strong co-repressor. It's not only important, I think, probably for development, but also with respect to defense. So there was a couple nice papers that came out last year on REL2 and REL2-like proteins that are important for, I think, defense and immunity for like Ustalago and a couple other fungus. So yes, absolutely. It seems to be very well conserved in that way. It's not the same sort of phenotypes though. So like the REL2 mutant, it's called Ramosa enhancer. These Ramosa mutants have these branching defects, especially, but it's not the same kind of transition of apical sulfate like during embryogenesis. That doesn't seem to be the case actually. So yeah, molecularly it seems to be the same though, but the phenotypes manifest differently. Yeah.
I have a more general question with regard to comparing morphogenesis and regeneration. Would you say that the networks would be relatively similar?
I would think so. That would be something that would be cool to actually parse out. But yeah, I mean, if we are capturing essentially what is a developmental trajectory, right, then yeah, they should be, because through that generation process, there's going to be differentiation and yeah. It's so bizarre. It's even more bizarre because we actually are keeping stuff going in both Arabidopsis and maize for pills 2 and pill 6. So we've put the maize pills 2 and pill 6 into the respective Arabidopsis mutants to see if they can complement. But we also just created the pills 2-6 double mutant in Arabidopsis. So the parental lines are big. The double mutant is so small. It's very, very teeny. So it's like the opposite of hybrid vigor, but it would be the phenotype I would have predicted to see based on the maize genes. Those are tDNA lines in Arabidopsis, and we've only made the combination with one allele of each gene. So I'm hesitant to really like make any strong conclusions yet, whether or not it's like synthetic lethality or what. I think we need other alleles. I mean, these tDNA alleles in Arabidopsis can be a real pain in the ass because they're not always clean backgrounds. So yeah, I don't know. So I don't know if it's a divergence thing in terms of different protein associations. I don't know yet.
Kind of a wake-up call in terms of also translation.
Yes. Right, but no, yeah, it's absolutely like the opposite, especially because the pills, you know, like if you were to try to find genotypes that have reduced expression of pills 2 or pill 6, you'd have a bigger plant maybe, right? You'd have increased yield, like things that seem to be goals that most folks want. But in fact, that would probably be the opposite. Especially like the pill 6 plants. I mean, they're very short. Well, W22 is short anyways, but they're very, very short. They're somewhat chlorotic. I mean, they're pretty crappy plants. So that definitely is not something you want to breed for in terms of a benefit. Yeah. I agree. Yeah, you got to just do it in the real deal context. Yep.
Kind of related to that, if you spray Auxin herbicides on qualities and grass, totally different. You have insight into why that is?
That is like the holy grail question. Lots of people want to know that. I don't. I mean, I think there's got to be something going on. So like we've done these transcriptional studies with IAA, and obviously like there are genes that are shared, like with Arabidopsis, right? Like we compared our study with work that Bus had done in the root to see like what genes are shared, but there's a lot of things that are totally different. So does that mean that the Auxin signaling module is there, but it's somehow rewired in terms of its output so that like when you're spraying gobs of IAA, like the grass just doesn't care, but other plants are very sensitive to it? I don't know. One of the receptors in maize, the AFP2 maize receptor, apparently is like hypersensitive to auxin. So at very, very low concentrations, it can induce degradation of oxidase like very quickly. It's like the most sensitive of the receptors. So to me, that's like the opposite of what I would, you know, that they should be hypersensitive to auxin, but in fact, they're insensitive. I don't know. A lot of people are still trying to figure that out of why. We wanted to do some more gene expression stuff, not with transcriptomics, but actually with protein, with proteomics, where we could maybe hit corn seedlings with different auxinic compounds and then try to look to see what's actually changing and what's unique, and do the same side by side with like soy or something like that. Yeah. Is there another question? Yeah.
Very nice talk. You mentioned hybrid breeding. You compare, you integrate hybrids, for example, P73 in combination with Dior 17, very successful. So if you have in your channel P73 and Dior 17,
yep,
look at the hybrid to see. What they do with hybrid.
Yeah, we haven't done any of those like with a QTL type approach. We do have the Mo 17 B73 real population actually from another project that my husband's been working on for defense. But we do have those genotypes in there. And then actually another thing we have been doing is introgressing our mutant alleles from W22 into things that make good hybrids, like LH244, PH207, PH251, B104. We're still at kind of like back cross 3-ish, you know, one season a year, things are a little bit slow. So we will get there. And yes, I think it will be great eventually to look at now that we've altered these root systems, does it impact yield? Can we make hybrids with those that actually have some beneficial traits? So yeah, Sean Kepler, who's a kind of more, into yield and things like that, is someone that I've been working with now. So Nick Lauder, who was at USDA, was someone who I had started those experiments with, and then he died 2 years ago. And so like stuff had started, but it's not, it'll take a little bit more time, I think, but yes. Yeah. All right.
Yeah, please join me in thanking. Thank you all very much. The students and postdocs, there's an opportunity to have lunch. If you're stick around. We're going by.